Right now, some of the absolute most expensive, highly sought-after skincare and, well, regenerative medicine in the world relies on an ingredient that honestly sounds like a prank.
Yeah, it really does.
Right. I mean, if you spend any time looking at aesthetics trends online, you've probably heard those whispered rumors about salmon sperm facials. Or maybe you've seen something a little more sterile listed on a high-end serum like sodium
Which
DNA.
definitely sounds a lot better on a label.
Exactly. But turning raw male fish reproductive tissue into a universal human healing agent, it requires this molecular gauntlet that is so extreme, it is genuinely a masterclass in chemical separation. So, okay, let's unpack this. Today, we are taking you on a deep dive into the wildly complex world of PDRN, or polydeoxyribonucleotide.
It is quite the mouthful.
It is, and we have pulled a giant stack of highly technical sources for this. We're talking molecular biology patents, clinical literature on DNA extraction, and pharmaceutical manufacturing protocols. And the mission today is to show you how scientists isolate pure, pristine DNA while utterly destroying the immunogenic proteins that cause severe allergies.
Which is, the ultimate diagnostic and manufacturing muddy water because biology is inherently messy.
Right, it's not like working with a clean synthetic chemical.
Exactly. You are taking raw, complex animal tissue and trying to pull a single, flawless, microscopic thread out of it without bringing any of the biological baggage along. I mean, biology is full of overlapping systems, sticky lipids, and cellular debris. The real hurdle we are looking at today is exactly how to navigate that mess.
Because you have to strip away everything that makes a salmon a salmon.
Yes, until you are left with nothing but the fundamental mathematical code of life.
And before we can even talk about purifying that code, we really have to look at the source material itself. Why are scientists starting with salmon milt, which is the male reproductive tissue, in the first place?
It's a great question.
Because, I mean, if you were going to go through all this trouble, why not use something easier to access?
Well, what's fascinating here is the biological imperative of the salmon itself. Pacific salmon, like the chum salmon, which is scientifically known as Oncorhynchus cata, or even the rainbow trout, they essentially reproduce once at the very end of their life cycle.
Right, the big final run up the river.
Exactly.
Yeah.
Their entire existence culminates in this one single moment, so all of their energy is redirected toward reproduction. The milt becomes this highly specialized DNA carrier rocket.
A carrier rocket, I like that.
Think about a regular somatic cell in your body, or even female fish roe. Those are filled with complex organelles. They have mitochondria to generate power, massive amounts of nutrient-rich cytoplasm to sustain a growing embryo.
Lots of extra stuff.
So much extra stuff, but sperm cells have eliminated almost all of that. They're incredibly stripped down and just unbelievably dense with highly condensed DNA.
So if you are a manufacturer, you obviously want the raw material that gives you the absolute highest yield with the least amount of cellular junk to clean up later.
Precisely. It's an efficiency game.
But beyond just the sheer density of the material, there is a genetic compatibility reason we use salmon, right? I was reading this source on the biological key to regeneration, and it pointed out that the DNA of salmon shares over 90% similarity in protein coding sequences with human DNA.
It does.
Some of the metrics in the literature even put the base pair similarity as high as 96.5%, or even 98%.
Yeah, and that genetic overlap offers massive biological compatibility, especially compared to something like plant DNA.
Which we see all the time in skincare.
We do, but plant-based materials are rich in secondary metabolites. Those are unique chemical compounds the plant uses for its own survival and defense systems, and they're completely foreign to human physiology.
They're essentially alien to our bodies.
Right. Salmon DNA, however, operates within a genetic framework our bodies already intuitively recognize. In fact, human mitochondrial DNA shares a 64.1% structural similarity with Oncorhynchus mycis, the rainbow trout.
That is wild.
Because the base composition ratio, the adenine, guanine, cytosine, and thymine, is so highly consistent with ours, human cells can metabolize and utilize these degradation products with almost zero waste.
But to actually utilize it, you have to get to that pristine, greater than 95% pure DNA. And that means you have to get rid of the protein.
Well, the dreaded proteins.
Right, because if you inject or apply raw fish tissue to a human, you are asking for a severe, potentially life-threatening immune response. We are looking at proteins specifically like parvalbumin, which is the major fish allergen, and a protein called protamine.
And protamine is arguably the most stubborn hurdle in this entire process.
Really? Why is that?
Well, during spermatogenesis in the salmon, Protamines actually replace standard nuclear histones. They are these arginine-rich, highly positively charged proteins.
Okay.
Because DNA has a negatively charged phosphodiester backbone, these positively charged protamines bind to it incredibly tightly. They practically glue themselves to it.
Opposites attract.
Exactly. They neutralize the DNA's charge and cause it to condense into these microscopic, tightly packed toroid or donut-like structures. A single sperm cell can contain up to 50,000 of these toroids. It is basically nature's way of armor-plating the DNA so it survives the harsh ocean environment.
So I picture the raw milt like a tightly tangled ball of yarn covered in sticky burrs. The yarn is the valuable DNA we want to extract, but the burrs are the allergenic proteins holding the whole tangle together.
That is a perfect analogy.
And if you are allergic to fish, those burrs are exactly what trigger an IgE-mediated allergic reaction. But wait, if the protamines are wrapped that incredibly tightly around the DNA to act as armor, shouldn't ripping them off destroy the DNA entirely?
You think so?
It just seems physically impossible to separate them without breaking the delicate DNA backbone we were trying to harvest in the first place.
And that is the exact problem early researchers faced. You absolutely cannot separate them while they are locked in that toroid structure. So the first step of the manufacturing gauntlet relies on extreme environmental shifts to force the structure to open on its own.
Okay, so how do they do that?
One of the primary extraction protocols involves a massive influx of salt. You take the diced testes and blend them with a pure 0.14 moles per liter sodium chloride brine to homogenize the tissue into a paste.
Making a salty fish paste.
Essentially, yes. And then you shock that paste with a high concentration 1.5 moles per liter brine.
So you are basically drowning it in an intensely salty ocean. Why does that make the armor fall off instead of just, making it saltier?
Because the salt chemically outcompetes the proteins, the brine floods the environment with positively charged sodium ions and negatively charged chloride ions.
Oh, I see where this is going.
Right. These ions swarm the toroid structures and begin interacting with both the positively charged protamines and the negatively charged DNA. The sodium ions essentially steer the DNA's attention, and the chloride ions distract the protamines.
So they just let go of each other.
Because of this massive chemical interference, yes. The tight electrostatic bonds holding the donut shape together collapse entirely. The DNA chains just unravel and unspool into these long, floating strands.
Okay, so the yarn is finally unspooled, but those allergenic burrs, the protamines and the parvalbumin, they are still sitting right there in the vat.
Floating right next to the DNA.
Right, so we need specific targeted methods to tear down those proteins without damaging the newly exposed DNA. And looking at the sources, in the past, scientists used liquid partitioning with organic solvents like phenol and chloroform.
Which is terrifying by modern standards.
Yeah, because liquid partitioning works by dissolving the proteins in a solvent that separates from the water-based DNA solution, much like oil separating from vinegar. It was highly effective for pulling proteins away, but it was declared a severe health and environmental hazard.
Phenol and chloroform are extremely toxic. They are highly flammable, corrosive, and they are known carcinogens.
And if you are manufacturing modern pharmaceutical-grade PDRN to, say, inject into a human joint or apply to an open wound.
Leaving behind even a trace amount of carcinogenic solvent residue is completely unacceptable.
Right, so the modern pipelines use food and pharma-grade methods instead. The sources detail how, once the DNA is unspooled in that heavy brine, a potent 0.5% elution agent is pumped into the mixture. Specifically, an SDS solution sodium dodecyl sulfate.
Which is brilliant. SDS acts as a chemical crowbar. It is a powerful anionic detergent.
Detergent like soap.
Very similar, yes. When it hits the unspooled mixture, it forcefully binds to the allergenic protamines, imparting a negative charge to them. This causes the proteins to violently repel each other and unfold, pulling them entirely away from the DNA chains.
Oh, wow.
But simply peeling the proteins away with detergent isn't enough. The unfolded proteins are still physically present in the solution. You have to physically chew them up into harmless fragments so they cannot reassemble or trigger an immune response.
Which brings us to the enzymes. The manufacturing data lists several very aggressive protein-eating enzymes used in this stage. Things like proteinase K, alkylase, aminopeptidase, and keratinase.
Molecular heavy hitters.
Yeah, but here's where it gets really interesting to me. If we are dropping strong detergents and aggressive, basically flesh-eating enzymes into this soup, why doesn't the DNA just get shredded along with the fish allergens? You've built an incredibly hostile chemical environment.
It comes down to structural targeting at a microscopic level. Those enzymes are highly specific molecular scissors. Proteinase K, for example, is engineered by nature to cleave peptide bonds.
And peptide bonds are.
They are the specific chemical bridges that link amino acids together to form proteins. DNA, however, does not contain peptide bonds at all. Its backbone is held together by phosphodiester bonds.
So the enzymes literally cannot see or interact with the DNA.
Exactly. They gorge themselves on the cellular debris, the parvalbumin and the protamines, slicing them into tiny, harmless peptides, and they completely ignore the fragile nucleic acids floating right next to them.
I'm trying to picture this vat right now. We have unspooled the DNA. We've broken down the fish allergens into tiny fragments using enzymes. But now we have a completely new problem. We have a soup of pure DNA, shredded fish allergens, and the very enzymes we just added.
Yes, we do.
And those enzymes are proteins themselves. If we want medical-grade purity, we need to get every last trace of protein out of there. My first instinct here, honestly, is just to boil it. Thermal sterilization. You heat the solution up to 100 degrees Celsius, the proteins denature, and you're left with clean DNA.
Heat inactivation is a very common thought process because, frankly, it is simple and cheap. And it does actually work for some non-specific enzymes like pancreatic denase I.
Okay, so why not use it?
Because relying on boiling is fundamentally flawed for producing high-purity PDRN. First off, if there are any calcium or magnesium ions present in the solution, they actually shield the enzymes and interfere with the thermal inactivation.
They act like little heat shields.
Exactly. Second, some enzymes are incredibly heat-resistant. Taq DNA polymerase, which is frequently used in lab settings, thrives in high heat and is nearly impossible to permanently destroy just by boiling it.
And even if you do manage to denature the enzymes with heat, the sources bring up a terrifying concept called reactivation.
Ah, yes, the zombie enzymes.
Right. You boil them, they look dead, but once the solution cools down, they just fold back up and start eating things again.
Furthermore, exposing DNA to high heat for extended periods causes depurinization. The heat literally starts breaking the bonds between the purine bases, the adenine and guanine, and the DNA backbone itself.
Oh, so you end up degrading the very product you were trying to save.
Precisely. Plus, boiling doesn't physically remove the shredded proteins or the dead enzymes from the solution anyway. It just leaves a floating graveyard of unfolded proteins mixed in with your DNA.
So boiling actually wrecks the product and leaves all the debris behind. What is the alternative then? This is where we get into a brilliant piece of biotechnology detailed in U.S. Patent 9,079,157b2.
The silica patent.
Yes, it outlines a cutting-edge deproteinization method using a specialized protein removal region.
If we connect this to the bigger picture, this patent solves the problem by moving away from trying to simply kill the enzymes with heat and instead focuses on physically trapping and dragging them out of the solution entirely.
Dragging them out physically.
Yeah, they introduce a solid phase extraction material. Specifically, they suspend cyanocropyl derivatized silica in a buffer containing dicarboxylic acids, salts like adipic acid or sabaric acid, at a highly controlled pH of around 5.0.
Whoa, back up. You completely lost me at cyanopropyl derivatized and dicarboxylic acids.
Yeah.
You are basically just reading the patent at me right now.
Fair enough, fair enough.
I need to know what is actually happening in the vat. How does this chemical mixture grab the proteins without grabbing the DNA? Explain it to me like I'm 5.
Let me simplify the chemistry. Think of the silica spheres as microscopic heavy rocks. Normally, these rocks wouldn't do anything at all, but the scientists coat these rocks with a very specific chemical adhesive. That's the cyanopropyl and the acids.
Okay, rocks with adhesive.
This adhesive uses two forces to trap proteins. First, it uses an electrical charge to attract the proteins, pulling them toward the silica rock. Second, it chemically attacks the water surrounding the protein.
It attacks the water.
Yes. Proteins hold their three-dimensional shape because of how they interact with water. The adhesive on the silica violently disrupts that water network, which forces the protein to lose its shape, uncoil, and permanently stick to the rock.
Okay, I absolutely love this. It's like throwing a highly sterilized microscopic lint roller into the vat.
A molecular lint roller.
Yes. The silica spheres are the roller, and the chemical coating is calibrated to be incredibly sticky, but only to proteins. It magnetizes them, breaks their structure, and permanently glues them to the roller.
And because this uncoiling and gluing process involves multiple points of contact across the entire protein molecule, it is absolutely irreversible.
So no zombie enzymes.
Exactly. The patent data shows that even if you leave a protein glued to the silica in conditions that normally favor reactivation for over 90 hours, absolutely zero enzymatic activity returns. It is locked away forever.
And the most crucial part for our purification process, that microscopic lint roller has zero affinity for DNA. The chemical adhesive doesn't recognize the nucleic acids at all, so the delicate DNA chains just float right past it entirely unharmed.
Once the lint rollers have swept up all the remaining proteins and enzymes, you simply centrifuge the mixture.
You just spin it.
You spin it at high speeds. The heavy silica spheres, which are now coated in the deactivated, tightly glued proteins, drop to the bottom of the vat like a heavy pellet. You then gently pour off the crystal clear liquid resting on top, which contains your pure, untouched DNA.
That is elegant.
It is. The patent notes that using just 2 microliters of this reagent can remove enzymes down to quantities that are completely undetectable by standard lab tests.
That is just phenomenal. So we've used heavy salt brine to unravel the armor, we've chemically stripped the proteins with SDS detergent, digested the fish allergens with molecular scissors, and used a derivatized silica lint roller to permanently extract every lingering enzyme. We are left with incredibly pure DNA.
But we aren't quite done.
Right, because raw salmon DNA, when fully unschooled, is about 2 meters long per cell. That is a massive, unwieldy genomic encyclopedia.
A 2-meter-long molecule is far too massive for human skin cells or joint tissues to absorb or utilize effectively. I mean, if you just applied that to a wound, it would sit on the surface and do absolutely nothing. It needs to be carefully sized.
Right, we can't just rub 2 meters of DNA onto our skin. So the manufacturers use specific nucleuses to intentionally chop the purified DNA into short-chain polymers. We are talking about turning that massive encyclopedia into highly targeted, easily readable excerpts.
Exactly.
Typically, pharmaceutical-grade PDRN is fragmented into sizes between 50 and 1,500 kilodaltons, which translates to roughly 50 to 2,000 base pairs.
And this enzymatic hydrolysis step dictates the entire medical functionality of the final product. If the DNA fragments are too large, they just can't penetrate the tissue. But if you chop them too small, they break down in the body way too quickly to stimulate the adenosine A2A receptor.
And those receptors are the goal.
Right. Those specific cellular receptors are the exact trigger points that command the human body to start building new blood vessels and reducing inflammation. The fragments have to be that exact Goldilocks size.
And to guarantee that perfect sizing and to provide one final physical barrier against contamination, the entire solution goes through ultrafiltration. The specifications of these filters matter immensely.
They really do.
According to the manufacturing protocols, top-tier pipelines force the gel through polyether sulfone filtration membranes with a 50-nanometer pore size, using booster pumps running at exactly 0.2 megapascals of pressure. Why that specific pressure, and why 50 nanometers?
The pressure is critical because PDRN at this stage is a highly viscous, very thick gel. If you push it too softly, it clogs. If you push it too hard, meaning exceeding that 0.2 megapascal limit, the sheer physical stress will literally tear the DNA chains apart, ruining your precise sizing.
It just rips them up.
Yeah. And the 50-nanometer pore size acts as an absolute physical barricade. It ensures that any rogue, inappropriately sized DNA chains, basically anything over 1,000 kilodaltons, and any highly resilient viruses or large molecular impurities are entirely blocked from passing through.
I noticed the sources contrast that with other methods that use ceramic tubes with a 20-nanometer pore size. If smaller is better for blocking impurities, why is 20 nanometers a problem?
Because a 20-nanometer filter is simply too restrictive for this specific compound. It drastically shifts the molecular weight distribution, filtering out the larger functional PDRN chains.
Oof, so you lose the good stuff.
You do. And this causes a massive drop in the viscosity of the final product. Instead of a thick, slow-absorbing gel that provides sustained receptor stimulation, you get this watery fluid that spreads too rapidly beneath the skin and is metabolized before it can actually induce proper tissue regeneration.
So what does this all mean for you, the listener? Whether you are looking at advanced wound healing for diabetic ulcers, severe joint regeneration therapies, or those high-end cosmetic applications, this grueling purification gauntlet is why these products can be used safely. The final PDRN ingredient isn't fish anymore. It is a greater than 95% pure nucleotide polymer.
There are 0 reproductive cells left, 0 cellular debris, and absolutely 0 fish odor.
Because the allergenic parvalbumin and the stubborn protamines have been completely dismantled by enzymes and physically locked away by silica, the risk of an allergic reaction, even if you have a documented severe seafood allergy, is practically non-existent.
Your immune system doesn't mount an attack because it simply doesn't see a foreign invader. It just sees the basic universal building blocks of life.
It is a remarkable testament to modern separation science. We are taking a natural biological byproduct and refining it through thermodynamics and physical chemistry down to its most fundamental
It
sequence.
really is.
Just to recap this insane journey, we started with salmon milk, an incredibly dense reservoir of DNA wrapped in tightly bound protein armor. We forced that armor to burst open using high-concentration salt brines. We chemically stripped the proteins with heavy detergents. We shredded the resulting fish allergens with highly targeted enzymes. We avoided the pitfalls of boiling by trapping those enzymes with an advanced silica adhesive. And finally, we cleaved the pure DNA into perfectly sized fragments and pushed them through microscopic 50-nanometer barricades.
You know, this raises an important question.